Hello and welcome to the Science of Childhood Cancer. Today we are privileged to have Matt Vanderheiden presenting from MIT. Before we get started, a couple of things. First, next week we're featuring the first in our series of Rising Stars of Cancer Research. These are shorter lectures from emerging scientists, emerging experts in the field. There'll be two 20-minute talks in the area of immunotherapy and chimeric antigen receptors from Yvonne Chen at UCLA and Sar Gill at UPenn. For questions during Matt's talk, please enter your question in the chat for all panelists. At the conclusion of Matt's talk, we will formally end at the top of the hour, but we will continue discussions for as long as people are engaged. So stick around after the talk and we will get to all of the questions that you put into the chat. Remember, please enter them for all panelists. So it's my pleasure to introduce Matt Vanderheiden. He's the director of the Koch Institute for Integrative Cancer Research and the Lester Wolf Professor in Molecular Biology at the Massachusetts Institute of Technology in Massachusetts. He's also an institute member of the Broad Institute of Harvard and MIT and an instructor of medicine at the Dana-Farber Cancer Institute and the Harvard Medical School. Matt received his MD and PhD degree from the University of Chicago and did his PhD under the mentorship of Craig Thompson. He completed clinical training in internal medicine and medical oncology at the Brigham and Women's Hospital and Dana-Farber Cancer Institute prior to completing a postdoctoral fellowship at Harvard Medical School with Lou Cantley. His laboratory studies how metabolism is regulated to meet the needs of cells in different physiological situations with a focus on understanding the role of metabolism in cancer. I've known Matt since he was a brilliant graduate student, and his continued contributions to the area of the metabolism of cancer have been startling, important, and groundbreaking. And some of those new findings he'll share with us today. Matt, I will turn it over to you.
All right. Thank you so much, Doug, for that extremely kind introduction. So, first of all, I just want to thank you, Doug, as well as everyone for inviting me to speak in this forum. It's really great to see people. And I, as Doug said, hope I can share with you today some of our recent work to try to understand what metabolic changes in cancer mean and how those relate to things that patients and physicians really care about. That is, how we might better treat cancer, as well as things like diet, which I will get to later, something very important to patients. And I actually will end with some very new stuff that we've been thinking about related to metabolism and cell state, just sort of following an introduction or the invitation that asked if I could talk about anything in that area. And we have stuff there that's actually one of the first times I will be presenting some of our very early stuff there. It's unfinished. And so I'm very much hoping that perhaps I can get feedback from you guys on ideas and places we can take that next. So just to get started here are my disclosures. So one thing that I always like to start off by saying is that metabolism is altered in cancer, and we know that this is true, and it's true in real patients because this clinical test, an FDG PET scan, works. Now, I'm sure most of you are familiar with this test. It's used frequently in the clinic as a tool to primarily stage cancer, sometimes look at therapy responses. But I like to remind people what an FDG PET scan is, and it's simply fluorodeoxyglucose. So it's glucose missing its two prime hydroxyl here on the ribose on the glucose molecule. This is replaced with the positron emitting F18 atom. This can be taken up, phosphorylated, cannot be metabolized further, and that positron emission from the F18 can be read out on a PET scan, which when fused to a CT scan gives you an image like this. And effectively, what this tells us is it basically is a way to look for who's taking up glucose in the body and how much is taken up. And it tells you that these tumors here are taking up more glucose than the surrounding normal tissue. Now, exactly what this PET scan means is something that I think many of us could debate. However, most people really assume that this has something to do with this very old observation in cancer biology, this so-called Warburg effect, which is arguably, we're coming up on the 100th year anniversary, one of the oldest differences noted between cancer and normal tissues. Now, I suspect many in the audience are also familiar with this experiment. This was Otto Warburg, who is a German chemist who was following up on the findings of Louis Pasteur, who of course described fermentation in microorganisms and that oxygen affected how much fermentation microorganisms do. And the experiment Warburg was carrying out was really to ask how true was this in mammalian tissues. And he found that most tissues behaved like Pasteur's yeast, that is, how much oxygen was present determined how much fermentation happened. But cancer was somehow different. It tended to do a lot of fermentation regardless of how much oxygen is there, a phenomenon known as aerobic glycolysis. He also described that the cancer cells took up more glucose, maybe the precursor for the FDG PET scan. And in the end, when you ask most clinicians why this happens, that is, why are tumors FDG PET positive? Well, the typical answer is that, oh, well, cancer needs more energy to grow, and that's why it takes up more glucose. And of course, that might have something to do with it, but there's something somewhat unsatisfying about this when you compare it with Warburg's observation of aerobic glycolysis. And that's because you don't have to remember too much of your undergraduate or medical school biochemistry to remember that you get a lot more ATP by oxidative pathways than you do from fermentative pathways, really creating somewhat of a paradox. That is, why is cancer doing this less efficient, at least in terms of ATP production, if it needs more energy to grow? Now, I wish I could tell you why cancers take up so much glucose and why they use it. We think we understand why they do aerobic glycolysis, but we really don't understand glucose metabolism. But I think what I can tell you is that at least one way that we've conceptualized some of these findings, which is sort of will set off what I want to talk about today, is just reminding you that energy comes in lots of different forms. And that is, ATP is certainly important for all cells. It's the currency that allows cells to, favor otherwise thermodynamically unfavorable reactions. And this is something all cells have to need to do, including proliferating cells. Non-proliferating cells have to do this just to exist as cells and carry out their normal functions. And yes, you need a little bit more ATP to proliferate, but what surprises most people is that the fractional increase in ATP production that's necessary to undergo cell proliferation is quite small relative to how much ATP you need just to exist as a cell. But rather, most of the energetic differences between proliferating and non-proliferating cells, or if you want, cancer and normal, comes down to this, and that is you need energy in a different form to proliferate. That is, in order to proliferate, you have to duplicate all of your biomass, which means you have to somehow acquire a whole bunch of stuff, proteins, nucleic acids, lipids, all the stuff that's necessary to go from one cell to two cells. And one way that we and many other people in the field have been thinking about whatever your favorite metabolic change in cancer is, be it FDG PET scan or aerobic glycolysis, is likely telling us not just about how the cells are getting ATP, but rather how those cells are repurposing their metabolic program or adapting their metabolic programs in a way to now carry out this new function. This is a function to not just make ATP, but rather make all the stuff that's necessary to go from one cell to two cells. Now, of course, a lot is known about this. People have been studying metabolism for decades, and it's well described how you can start with your favorite nutrient and turn it into all of these components. But one thing I like to remind people of is that, most of our cells in our body are never in a nutrient-deprived state. That is, there's really no evidence cells are ever ATP starved. And in fact, there's tons of nutrients around. But if we go back to that FDG PET scan, remember, there is this big difference in just what cells or what tissues in the body are using glucose. And so I would argue a big question isn't just, if we want to understand these phenotypes, it's really asking the question, why do specific cells, cancer or normal, different tissues or cancer from different tissues, how do they decide which nutrients in their environment to take up and how do they allocate those to different places? And I think if we could understand that question, I think we could go a long way to better understanding exactly what these different metabolic changes mean, why they're different from cancer to cancer. And ultimately, I would argue it tells us something about therapy. And part and parcel to this is sort of the notion that not all cells do the same metabolism and not all cancers do the same metabolism. And I think we can say that with confidence simply because there's another clinical observation, and that is that our chemotherapy drugs work, and they work differently in different cancers. Now, chemotherapy kind of has a bad name these days. We usually don't give a talk about new drug development and talk about chemotherapies. But the bottom line is, and the clinicians certainly realize this, is that we still give a lot of chemotherapy to patients. And we do this because it works, because these are powerful drugs that contribute to cures in some patients. But these drugs don't work in all people. Now, this notion that these are nonspecific poisons that attack all dividing cells is simply not fair. Many of our chemotherapies are quite targeted, and many of them are actually quite targeted at enzymes and nucleotide metabolism. Take, for example, a commonly used drug. So I'm an adult oncologist, not a pediatric oncologist, but 5-FU is a very powerful drug. It is curative as adjuvant therapy for colon cancer, yet it does not work in melanoma. There's similar oncogenic mutations across those two different diseases. There's similar proliferation rates. Yet 5-FU has a clear target. It's thymidylate synthase. You can't argue that thymidine is not needed by melanoma cells or colon cancer cells. Yet the fact that 5-FU works differently in one cancer where it's quite effective and in another cancer is not effective at all, to put it in genetic terms, really argues that you have differential sensitivity to thymidylate synthase inhibition, or to put it another way, differential sensitivity to an essential gene in metabolism. Now, I think if we could understand that differential essentiality to these essential genes, that would tell us a lot about how to use our therapies more effectively, what other therapies might be out there. But it also tells us that those two cancers must not do metabolism to grow in exactly the same way, or they wouldn't have this differential sensitivity to exist in the first place. And so we've been thinking about this a lot, and we've been thinking about if you'll accept that not all cancers do the same metabolism. Well, then the right question becomes, well, what determines how different cancers do metabolism differently? Because that must underlie somehow how to select patients for this or how to select metabolic targets if we're going to use this to treat cancer. And I want to summarize a lot of work here simply in a single slide. This is work from us as well as many people that contributed to this. When we thought about, well, if not all cancers do the same metabolism, what determines how they do metabolism? And of course, you know, if you want to ask what makes different cancers different, you have to start with the genetic mutations. It's very clear that different oncogenic mutations cause cancer. And those mutations differ across different tissues, contribute to a lot of phenotypes, including metabolism. Work from many labs have shown that specific oncogenic driver lesions can have effect on metabolism. However, it's also very clear that these mutations act within the context of where a cancer comes from, particularly with respect to metabolism. There's actually work that we had a minor contribution to years ago that's now been replicated by many other people that I think asked a very interesting question. And it simply asked at the gene expression level, if you look at metabolic enzymes, how similar are cancers to themselves versus their normal tissue counterparts? And what was quite surprising is that just from a metabolic gene expression standpoint, you can say where cancer came from. That is, it looks much more like the tissue, the cell of origin, the lineage of that cancer, than it does like other cancers. Now, the way we've interpreted this is as follows. Of course, different normal tissues, brain, liver, heart, do different metabolism because they have different functions and they have a metabolic network, a metabolic gene expression that's adapted to carry out those functions. Now, it could happen that when you form a cancer in those tissues that you revert to some common metabolic program, but that's not what the gene expression data was suggesting. Rather, it was suggesting that you take the existing metabolic program of a tissue, alter it with the various genetic mutations, and this somehow creates a new metabolic program that now allows that cell to proliferate. Now, this might begin to explain how you can have differential sensitivity to essential genes. That is, cancers are not reverting to some known proliferative program, but rather taking something existing and cobbling it together so that you can now proliferate. However, I think there's an additional component to this and that is if you take whatever metabolic network and you now throw it into a situation like tissue culture media where absolutely no nutrients are limiting, well, that network is going to look very flexible, and you're going to, it's a very popular thing to talk about the flexibility of metabolism and why that may limit the effectiveness of targeting these pathways. However, the reality is that our cancers don't live in tissue culture media. They live in the environment of tissues. And so we would argue that it's really the constraints placed by the nutrients available in the tissue on the unique metabolic network that's defined by where the cancer came from and how it's altered by the genetic mutations that ultimately defines what are the metabolic programs that the cancers run, with the bottlenecks really lying at this intersection between the constraints placed on the environment, on the unique network of each cancer. And so we've been working really hard to try to understand what specifies the network of each cancer, that is how many networks are out there, as well as on this side, and that is how are different tissues constraining those networks as a way to actually define how different therapies might work. And a lot of what I want to talk to you about today is our recent efforts would have really been focused on this side of the equation, and that is how can we better understand what nutrients are available and what might that be telling us about the constraints that really exist for the metabolism of cancer. And so to begin to get into some data, I want to refer to a study that I'm just going to allude to briefly. It was published now 2 years ago, but this was work carried out by a former graduate student, Mark Sullivan, as well as a former postdoc, Alex Muir. Alex Muir now has his own group at the University of Chicago. And they really set out to define what nutrients are really present in the environment of cancer cells. And so they adapted some old programs to isolate interstitial fluid from cancers. So the fluid that's in the extracellular space, what arguably is available to the different cells in a tumor. And they wanted to know how that differed relative to blood, and they found it was different, so that was interesting. But they also wanted to know, by going back and actually keeping of those three variables, two constant and altering the third, how that actually affected nutrient availability. And so they took pancreatic cancer cells, put them in different places in the mouse, and found that different nutrients were available based on the tissue those cells the tumors were growing in. They took lung and pancreatic tumors that had the same driver mutations, put them in the same place in a mouse. So really asking how lineage affected this. That had an effect on which nutrients were available. But the surprise we found at the time is that we took two different lung cancers with two different driver mutations, put them in the same place, and we found that there was now much more overlap, which was actually quite telling because it began to argue that it was perhaps not that the cancer was defining its nutrient environment, but rather things extrinsic to the cancer were defining it. That is, either cells that were recruited into the tissue or the tissues that were present, and that the cancer really had to adapt to the environment that was found. And they also found, consistent with that diet, which could change nutrient availability, could also change nutrient availability in tissues, something I'll come back to later in the talk. And I think further evidence from this has really come from a collaboration that we've had with Kim Rathmill's lab. So, a couple hours down the road from you guys at Vanderbilt, because they were able to isolate and send us interstitial fluid from normal kidney, as well as kidney tumors from patients. And we compared that to what's in the blood. And so we did quantitative mass spectrometry, looked at nutrient availability, and just like what we saw in mice, what was found in the interstitial fluid of both the tumor and the normal tissue was very different from what it was in blood. But there was almost perfect overlap between what nutrients were just found in the kidney versus what was found in the kidney tumor, really arguing that something about the different tissues was what cancer had to adapt to, and really changing the way we started thinking about how nutrient availability might begin to relate to cancer cell phenotypes. Now, one immediate implication for this was maybe this was telling us something about metastasis. So it's been known forever that specific cancers tend to metastasize to basically stereotyped places that are not always anatomically defined. And it's really observations like this that have led to the seed versus soil debate and metastasis for a very long time. And there's plenty of evidence out there that things intrinsic to the cancer cell, the seed, can affect whether it can metastasize or not. But the fact cancer cells grow in these stereotyped places also said that maybe there's something about the soil that also affects metastasis. And the immediate thing that suggested is that if each tissue had its own unique nutrient environment, perhaps it was the ability of cancer cells to be able to access the right nutrient environment that somehow was contributing to the question of where cancer cells might be able to metastasize to. And this was an idea that we have, in my group, have found quite attractive for some time and have been studying for a while and have tested in a lot of different ways. And so here's 3 different postdocs who joined my lab to work on this. We do a lot of work in pancreas cancer. And so they would take pancreas cancer cells and look at primary tumors versus metastases or things implanted in the flank, all kinds of different places, and do all kinds of fancy metabolic phenotyping, including looking at the fate of, say, C13 labeled glucose. And I'll just summarize a lot of what for them is really painful data, and that is they kept doing these experiments, and they kept finding over and over again that the metabolism just didn't look that different regardless of where they put the things. Now, this is somewhat sad if you're a postdoc, because of course it's really hard to publish that things are the same. It's a lot easier to publish that things are different. And of course, there's work out there that argues special things to grow in different sites. And that is very, very good work. But the big picture was really telling us that maybe there might be more similarities than there are differences. Now, this was somewhat satisfying if one goes back and thinks about the clinic, because the reality is that if a patient has metastatic cancer and you give them a drug that they're going to respond to, they tend to respond in all sites. Now, you may get specific resistance in a particular site, but the fact that at least initially they respond in all sites really tells you something, that there has to be at least some commonality across those sites that allows those cancer cells to respond. And based on that, Sharanya Sivanan, a current postdoc in my lab, really took on this question about, could we really generate some evidence that at least might be consistent with this model, that similarities might be driving this common metabolic phenotype that we were observing across sites. And so she really took this seriously and took primary pancreas tumors as well as naturally occurring mets, and she started implanting these all over the mice into primary and metastatic sites, and was really quantitative about what actually happened. That is, were cells better able to grow in these different sites? So she wasn't looking at the ability to metastasize or not. The usual thing is, do your cancers form metastasis, or can you find a site? That's not what she was asking. She was asking, if she took out the ability to actually reach a site, were there differences in cancers to be able to grow in those different sites? And I think she found something actually quite interesting. And so, she could take cells from all these sites and they grew pretty similar in culture. So it turned out that wasn't going to be very helpful. But what she found is that no matter where the cells came from, they always formed big tumors in the pancreas. And we actually had a hard time really telling differences in terms of tumor growth rate or size of the tumors that formed. If you put the same number of cells and looked over time, how quickly you formed a tumor in the pancreas, or in any site for that matter. But you always formed big tumors in the pancreas. And yes, you could form tumors in these other sites, but in a quantitative way, those tumors were always much, much smaller than what we saw in the primary site. That's just shown here. So here's plenty of tumors would form in the liver, but these are tiny tumors, and the same number of tumor cells injected in the pancreas would give you these gram tumors over the same period of time, really consistent with this notion that even metastases still like to grow in the primary site better than they like to grow in the metastatic site. Well, Sharanya took this a step further, and she actually did Massaguet, or originally Fittler type experiments, where she actually selected for cells by repeatedly passaging them in vivo, such that they really were, preferred to grow in the liver or lung, at least in the classic assays about could you find the liver or lung. And when she did this, it actually had zero effect on this quantitative question about whether or not where you preferred to grow. even after this selective in vivo passaging to grow in the liver or lung, these cells still preferred to grow in the pancreas, at least in a quantitative way, even though, of course, they could form liver tumors or lung tumors or grow in the flank. They still always preferred to grow in the pancreas, really arguing that maybe something about growing in the primary site is something that's not so easy to select out of a cancer cell. We could do metabolic profiling in other ways. And so this is just shotgun metabolomics, just asking at a 50,000 foot view, how similar are the tumors to the normal tissues? And, the liver metastases and the primary pancreatic tumors cluster very well with the pancreas. Same thing with the lung metastases, even the selected lung metastases in primary pancreas tissue. And this clusters away from what you would see in normal liver or more normal lung, which again is consistent with this notion that perhaps you're adapting the metabolic program of the tissue you came from, and therefore you're always going to be best adapted to grow in the tissue where you came from, because that is the nutrients where that program is best fitting. I think the real test of this is now to move to a different tumor model and ask, well, is this transferable? Because a prediction would be is that if you're a pancreas tumor and you compare growth in the pancreas or lung, you're always going to prefer to grow in the pancreas than you do in the lung. Whereas if you're a lung tumor, well, it should be the reverse. You should prefer to grow in the lung than you prefer to grow in the pancreas. And we chose these two systems because they're very nice. We can do the same genetics to induce tumors in these two models. And so that takes that variable out of the equation. And in fact, we get exactly the predicted result. So pancreas tumors, even lung mets selected lung mets always grow better in the pancreas than they do in the lung. Lung tumors always grow better in the lung than they do in the pancreas, really consistent with this notion that perhaps, at least metabolically, you prefer to grow in the site where you actually came from. Now, I think the real thing that helps with support this hypothesis is really looking at the exception. And the exception is if I go back to what I told you earlier about where do cells actually respond to drug or fail to respond if you have a metastatic site. Remember, I said that if you have a metastasis and you're naive to a therapy and you respond, you tend to respond in all sites. Well, the exception to that is, of course, the brain. There's lots of examples of therapies that work well in an extracranial disease, but fail to work in the brain. A classic example of this is breast cancer, HER2-positive breast cancer. We can target HER2-positive breast cancer quite well and get good peripheral responses. Those things fail in the brain. Now, often this is viewed as a pharmacokinetic issue, and there is, of course, it's challenging to deliver drugs to the brain, but lots of studies from various people, including our collaborators on this project, Rakesh Jain, had shown that, something about being in the brain environment also, even if you get the drug there, affects your ability to respond to drugs, which really led us to begin to ask questions. Well, was there something different metabolically about growing in the brain than the other sites? And this work here was also published earlier this year, so I don't want to spend too much time talking about it. You can read the paper if you were interested. But effectively, working with Gino Ferrara and Rakesh Jain's lab, two of my students, Ahmed Ali and Alba Luengo, basically identified what the postdocs have been looking for, and that's a site-specific metabolic dependency to grow in the brain. And what they found is that to grow in the brain, you basically had to make your own fatty acids. The reason for this is because you can't get fatty acids from the brain. There's plenty of fat in the brain, but it's not accessible to the breast cancer cells. And so therefore you have to make them. And that this was dispensable in the other sites because there were plenty of fatty acids to take up from the environment in those other sites, really arguing that something about growing in the brain was different than growing in the primary site. Now, that's great, but now I want to come back to sort of a larger question that goes back to what actually drives this site-specific dependency. That is, why do you care? Why can't you just, turn up to make whatever you want, right? If you're in a location where, why do you ever care about which nutrients are available or matching your network to available nutrients in a tissue? And because, a fair question would be, well, metabolism is adaptable. Can't you just go to the brain and, sure, if there's no fat there, turn on fatty acid synthesis? Well, the reality is those experiments were telling us that not all cells can go to the brain and turn on fatty acid synthesis. You had to do that to form a brain metastasis, but only some cells could do it. And the same thing with those pancreas and lung transplantation experiments. You still always grew better in one site. Why can't you just turn on to make the things that you can't get from your environment? And the way we've been thinking about this is follows. And it turns out that all cells are really made of the same things, right? Even yeast cells have similar ratios of proteins and nucleic acids and lipids as any mammalian cell, normal or cancerous. And so the difference must have to do with, well, maybe metabolism can't make all things equally. That is, maybe you have a metabolic budget that says you can only allocate this to make some things, which therefore addicts you to making those things and also addicts you to taking up those things from your environment. The analogy I like to use here is it's expensive to live in Boston, so I pay a graduate student a certain amount of money. Well, they can go out and, they have to allocate that money and it has to make do for their rent and their food and their entertainment. Well, if I give them free meals any day, now they can afford a more expensive apartment. But the reality is that they adapt to their environment. They can't easily change their budget, so they really become addicted to this. And so we become very interested in, well, what this budget might be. And to understand what we think it is, I have to torture you with a little bit about how metabolism works. You probably learned this once upon a time and forgot it. But now we'll come back to what is ATP? Well, ATP is basically you charge up a ratio of ATP to ADP, and that ratio allows you to carry out thermodynamically unfavorable processes. Well, what allows you to do that is that it turns out nutrient oxidation, that is removing electrons from carbon and putting them on something else, is favorable. Burning wood, burning gasoline, whatever, bodies do the same thing. And there you can couple those favorable oxidation reactions to making ATP. This is why things like lipids, reduced carbon, is calories, right? It's how we get energy to grow. And what you also learned is that what makes this work is that you have to dispose of those electrons somewhere. They have to go somewhere, some electron waste. And this is why you consume oxygen, or this is what fermentation is, because this is basically the electron disposal that allows you to do nutrient oxidation. Well, what we keep finding over and over again is that the things that are hard for cells to make have to do things like the amino acid aspartate, which is required to make proteins, or purine and pyrimidines and nucleic acids. And these things are all more oxidized than the nutrient cells consume. And so we think it's this necessity. Remember, all cells have this fixed or higher ATP requirement. So you have this fixed requirement to make ATP. But now if you're going to proliferate, you have this additional demand that you have to keep making ATP, but now you have to make all this oxidized biomass. You have all this extra electrons to get rid of. And it turns out that we think disposing of all these electrons now becomes swamped out, and that this ultimately is what becomes limiting for cells, and perhaps may also be what actually drives aerobic glycolysis, something I don't have time to get into in today's talk just because I want to focus on other things. But the hypothesis is that it's this electron disposal, this NAD availability, that then becomes limiting for cells. And if we look and ask, well, what do you need NAD to make? Well, we did a nice computational project with Dennis Vidkup's lab at Columbia University, where he simply asked on a theoretical basis, what's the NAD plus cost? That's it, is the oxidation cost of making biomass. And there's a whole bunch of things you need to make. And now you can begin to see, well, making these things all require NAD. If you get some of them from your environment, you have more NAD to make the other things. And you might say, well, I showed you about the brain and lipids, but even lipids has an NAD cost to make. Happy to talk about this in the question and answers. But it turns out there's even an NAD cost to make some things that you might think of as reduced biomass. And so we think it's really this limitation for NAD that ultimately leads to this model where NAD, your ability to do oxidation reactions, are the budget. And you have to save your ability to do oxidation reactions to make the things that you can't take up from your environment. So therefore, you care about making those things. But it also addicts you to having to take up other things from your environment. And this may underlie, at least at a hypothesis place, where you get this seed and soil difference for why cancers prefer to grow in specific nutrient environments, and actually maybe why our drugs actually begin to work differently based on where cancer comes from and where it's actually growing. Well, that's a great idea. Is there any evidence to actually support this? And I think we still have a ways to go to prove this model. But I think one thing that begins to describe what this might be, actually, I can use by thinking about the study that we, again, published 2 years ago with Tyler Jack's lab, where two of his postdocs, Lian and Shen Rong, did an experiment where they were basically asking for what was differential dependencies of small cell lung cancer versus non-small cell lung cancer and pancreas cancer. and identified that small cell lung cancer was more sensitive to this drug, Brequinar, or DHODH, which is basically pyrimidine synthesis, than non-small cell lung cancer or pancreas cancer. So effectively identifying that small cell lung cancer cares more about making pyrimidines than non-small cell lung cancer or pancreas cancer. That's basically what this says. And it's the embodiment of this differential essentiality on essential genes. Here you have two different cancer types. One cares about making pyrimidines more than the other ones. What's actually driving that? Well, at least part of the explanation actually came from my postdocs. Really, Chris Nabel really was key in helping to figure this out. And basically, what we published, as described as the explanation, is it really didn't have anything to do with the ability to make pyrimidines. It had more to do with differential propensity to salvage pyrimidines. And effectively, what we were able to argue is that non-small cell lung cancer and pancreas cancer was better at salvaging pyrimidines than small cell lung cancer. And what really sealed this is we could go to a different cancer, prostate cancer, which was also not very good at salvaging pyrimidines. And it was also sensitive to inhibition of pyrimidine synthesis. And so the argument is, well, maybe prostate and small cell lung cancer cares more about making pyrimidines because it salvages them less well. And non-small cell lung cancer and pancreas cancer cares less about making pyrimidines because it likes to salvage its pyrimidines. Well, that's cool. But the oncologist here, particularly if there's anyone who knows adult oncology, will recognize something quite interesting about this observation. And that is we have a lot of chemotherapies that are absolutely dependent on pyrimidine salvage, including 5-FU and gemcitabine. These are very effective drugs in non-small cell lung cancer and pancreas cancer. In fact, they are the standard of care in these diseases. But these are not good drugs in small cell lung cancer, and they do not work in prostate cancer. And so perhaps one of the reasons why this is the case is that these cells are more dependent on pyrimidine salvage, and that's why drugs that target pyrimidine salvage actually work in those cancers and don't work in these cancers, but these cancers are more dependent on pyrimidine synthesis because they don't salvage pyrimidines. They have to make their own pyrimidines, and really may begin to speak to this model we have where it's really how cells match what's in their environment to their metabolic network and how that applies to this budget to make different oxidized biomass components. And we're at least excited by the possibility that this may underlie some of this differential essentiality to different metabolic enzymes or pathways, and is something we've been really excited to test. Well, the next thing I want to talk about, which I think might be of interest to many people here because it comes up in my life a lot, is really an additional extension to this. And because that is, well, there's different ways to solve the budget problem. And we've been thinking about that, what actually defines the budget and whatnot. But to go back to my analogy about grad students get paid a certain amount of money and they have to allocate to all those things. Well, the grad students would just come back and tell me, well, just pay me more and now my budget will go further. Or another way to say it is, well, maybe we can change what nutrients are available and then that would change your dependency on the budget. And this is actually a way to think about something that really matters to patients, but we as doctors do not have an answer to, and that's diet, okay? Probably the most common question I get oftentimes from patients or their families who have pediatric cancers is, someone's been diagnosed with cancer, what should I actually eat? And I never have a good answer for them, and I think the world struggles with this question. But if we come back to this finding that I alluded to earlier, is that one thing that changes nutrient availabilities in tissues is simply feeding mice a different diet. Well, this led a current postdoc of mine, Evan Lien, who's actually going on the job market now. Evan is fantastic. And basically, Evan had the idea, well, maybe we could take advantage of this, because if diet changes nutrient availability, and diet can have different effects on how cancers grow, maybe we can correlate the effects of diet on phenotype with how it actually changes nutrient availability, either in the blood or tumors, and lead to new hypotheses and insights about how diet might actually intersect with the metabolism of tumors and therefore affect tumors growing. And he decided to apply this to glucose metabolism. He did this for a real reason, and that is because the most common question I get is, well, we know cancers take up a lot of glucose, and so therefore I'll just cut sugar from my diet, and if I cut sugar from my diet, I'll starve the tumors. Now, the people who have thought about this scientifically know that the relationship between glucose and tumor growth has at least thought to have more to do with insulin, But the naive patient idea cannot be right. And that's simply because, well, if you ask, well, what is glucose in blood or tissues? Well, in blood, it's 5 millimolar. We measure it in tissues to be well above 1 millimolar. This is still well above the KM for glucose uptake, or how glucose actually affects growth rate of a cell. It turns out the most extreme low glycemic diet you can go on, maybe you can cut glucose in half. That's somewhere in the 2.5 to 3 millimolar range, still well above how much glucose you need to take up. So you're still saturating. In fact, this is probably why FDG PET scan works. Again, it's an argument I will make. I don't really know if any tumors are ever at least on a bulk level glucose starved, diet or otherwise. Okay. But that still comes back to, well, how do low glycemic diets really work then? And Evan did a really interesting experiment where he basically took two different low glycemic diets. He took 40% caloric restriction, where he basically just took a standard mouse diet and cut the carbohydrates such that you had 40% of the calories. Or he went to a ketogenic diet, which is the most common thing that people usually ask me about. Should I eat a ketogenic diet if I've been diagnosed with cancer? And he did this, and we looked at a model of pancreatic cancer for a variety of reasons, although it turns out this is true in other cancers that he looked at as well. And basically what he found was that the caloric restricted diet was far more effective at inhibiting tumor growth than the ketogenic diet when he controlled for all things, even though both diets had similar effects on blood glucose and similar effects on blood insulin, which really argued that changes in glucose and insulin really can't be fully explaining the differential effect of these low glycemic diets on what's actually affecting tumor growth. And so to get at this more, he decided to do what we said earlier. Well, let's ask how these diets differentially change other nutrient availabilities. And basically what he found is lots of things changed, but the most abrupt thing, or the biggest changes he saw, had to do with fat. And this makes sense. He saw that there was a lot more fat in the blood and tumor interstitial fluid with the ketogenic diet than there was with the caloric restricted diet. In fact, these were quite depleted in the blood and interstitial fluid, and this has been known forever. The ketogenic diet is effectively all fat, and so it makes sense that fatty acids go up everywhere. And caloric restriction has been known forever that the biggest effect of caloric restriction is you lose all your body fat. And so this also makes a lot of sense with what is already known. When we were thinking about this separately, we were looking, well, what was the effect of actually take lipids away from cancer cells? And again, this also makes sense. If you take lipids away from cancer cells in culture, they turn on fatty acid synthesis. This has a number of effects. To turn on fatty acid synthesis, you have to make both saturated and unsaturated fatty acids. And it turns out that the way mammals, we can only make certain unsaturated fatty acids carried out by this enzyme, steroid-CoA desaturase. There's a couple other ones, but we're very limited in the unsaturation events we can do. And so basically, if you take lipids away and you have to make all of your own fat, you get depletion of polyunsaturated fatty acids, which are almost all derived from the diet. We can't make them as animals, so you have to take them up from your surroundings. And now you turn over and you make more monounsaturated fatty acids. Well, this ends up to be relevant because what Evan found is that it turns out steroid-CoA desaturase is an insulin-responsive gene. And so in the caloric-restricted mice, what he found is that you had lower fat, but you very specifically had lower ratios of unsaturated to saturated fatty acids. And we reason this is the case because insulin controls steroid-CoA desaturase expression. And so if you have the low insulin from caloric restriction, you get less steroid-CoA desaturase expression, and you now also get a perturbation. You not only your lipid depleted, but you also have a perturbation in your unsaturated to saturated fatty acid ratios. Well, this led then to a model for maybe what might be going on. Because the idea is that maybe the low insulin from the low glycemic diets decreases your ability to desaturate your fatty acids. And so now you also, when you have less fat, well, not only can you not make fat, but you also can't desaturate your fat. And perhaps this affects tumor growth. And in the ketogenic diet, where you had plenty of fat around, well, this is never an issue because you rescue the whole thing because there's plenty of fat from the diet. And so he decided to test this model in a number of different ways. The simplest thing was, well, let's just give them steroid-CoA desaturase back. And it turns out this had the ability to rescue some of the effects of caloric restriction, really arguing that this might be going on. Well, that's one possibility, but he wanted to test this from different ways. And he realized, well, if this was true, we should also be able to manipulate the fat content of the diet. And so the way he did this is he actually further lowered glucose availability. So he made a high-fat caloric-restricted diet. So this is an even lower glycemic diet, replacing some of the calories that are taken away from the sugar to now be more fat. And he could use fat from different sources that had different saturated and unsaturated fatty acid ratios. And effectively, what he found is that the soybean oil diet, high fat, so even though it was lower glycemic, still good at suppressing tumor growth, looked just like before, perturbed the unsaturated saturated fatty acid ratio, and that correlated very well. But the palm oil diet, again, lower sugar than the one before, but did not perturb the saturated unsaturated fatty acid ratio, now no longer has an effect on tumor growth, which is very consistent with this model that it's actually this alteration in fat saturation, unsaturation that's driving this in the setting of the low sugar and not the low sugar itself. We can prove this more because remember, ketogenic diet is low insulin too, and so steroid-CoA desaturases is low. And the idea is maybe it's the high fat though that's rescuing this effect. And we had made our ketogenic diets with lard because that's what a lot of people do, and lard is particularly high in the monounsaturated fatty acid 18-1, and so we really didn't see any change in the ratio of 18-1 to 18-0, the most abundant monounsaturated saturated fatty acid in cells. But then he asked, well, what if I reformulate the diet, not with lard, but with palm oil, which has much higher saturated fatty acid relative to unsaturated fatty acid than lard? And when he did this, what he found is that the palm oil ketogenic diet could raise saturated fatty acid levels and now began to show an effect on suppressing tumor growth, which was pretty cool because now this actually suggests something I think pretty deep about what might be going on about how to think about diet and cancer, both with these low glycemic diets, but also a way to begin to think about this with respect to, larger lessons about diet and how that might affect tumor growth. So the first one is, I think it suggests that the model is probably right, that at least one thing that controls whether low glycemic diet works is it's the low insulin that actually affects how you metabolize fat. And then depending on what your fat content is and whether or not that perturbs the saturated, unsaturated fatty acid contributes to whether or not the tumors can grow or not. But apart from that, if we go and actually look at the specifics of what we actually did to cause these different diets, well, it begins also to say that it's not that intrinsic fats are healthy or unhealthy. It's really the context in which those fats are given. So soybean oil turned out to be the right thing to do in a caloric restricted diet if you want to slow the tumor growth, meaning it was good for the patient, good for the patient, bad for the tumor. Whereas palm oil was the reverse. So you don't want to have palm oil in this setting because it's bad for you, meaning it helps the tumor grow, or at least the tumor is able to grow in this setting. But in the ketogenic diet, that's not what we saw. In the ketogenic diet, it turns out palm oil was the right thing to do because that helped the tumor grow less well. So good for the mouse, bad for tumor growth, whereas lard was the other thing to do. So it's not that palm oil is good or bad. It's in the context of how it's given and other dietary factors that also affects how tumor growth will be affected. And it also means that if we really want to understand how diet really is affecting cancer growth, we have to get beyond these sort of simple things like, oh, high sugar is bad or high sugar is good, because it's really going to be how that diet actually interacts with the unique metabolism of the tumor and other dietary factors that are ultimately going to drive responses, which I realize is more complicated, but may actually give us a role for a roadmap forward for how to better think about precision nutrition in cancer. All right, in the last 5 to 10 minutes or so, I want to come back to what I promised earlier, and that is you guys in your invitation letter to me said, well, we'd love to hear more about any thoughts you have on gene expression or cell state. And of course, it's been known for a long time that metabolism can influence cell state. There's a lot of beautiful work in this area, a lot of it in the immune system, stuff that I know, including a lot of beautiful work from St. Jude's. But if I can summarize a lot of data, how metabolism is influencing cell state largely comes down to a couple things. It usually has to do with things that perturb the alpha-ketoglutarate succinate ratios, which clearly affect epigenetic enzymes or create substrates for epigenetic modifiers. Yet the challenge of really understanding this is if metabolism, certainly it can work that way. There's clearly ways that can affect cell state, but really cell state differences have to do with how do you get specificity in gene expression? And it's unclear how you get to specificity by globally affecting, the activity of a, TET enzyme or a Jumanji enzyme, or changing SAM levels that might affect the activity of some methyltransferase. And so we've been thinking about this a lot. And in fact, there's two people in my lab who have been studying this. One is Peggy Sue, a former postdoc now at the University of Michigan. Another one is Brian Doe, a current MD PhD student. And I want to tell you a little bit about their work. This is not a finished story by any means, but I think it's telling us something quite interesting that is a different way for me to at least think about how metabolism might be affecting cell state. And this starts really following up on this observation. It was a publication in Cell now 5 years ago by David Sykes at MGH. He's been helping us with this as well. And basically, he reported that inhibiting DHODH, Brequinar, I alluded to this earlier, so pyrimidine synthesis, can cause differentiation of AML cells. And in this paper, he argues that it's not clear how it does this, but it probably doesn't do it via necessarily affecting uridine or nucleotide levels itself. Well, Peggy decided to ask this in a very unbiased way. So she took this system where she basically took the same model system that David Sykes had used in that paper. So this is a GMP-like mouse cell that's basically immortalized by HOXA9, constitutive HOXA9 expression. And basically, if you withdraw HOXA9 or do other things like Sykes did in his screen, you can basically see differentiation is read out by either CD11B expression or turning on a lysozyme GFP. And so Peggy decided to do a thing where she basically took a metabolic library that was designed by Isaac Harris. He's at Rochester now, but he was in Joan Brugge's lab at the time. And she basically did a drug screen focused on metabolism. And we've now done the same thing with CRISPR as well and got similar results, basically asking what could affect differentiation if we perturbed metabolism. And the results of these screens could not be more clear. And it basically told us that we can mess with nucleoside metabolism in all kinds of different ways, and it affects differentiation. We can see the turning on of these CD11B or lysozyme GFP occur at doses of perturbing nucleotide metabolism at doses that do not kill cells. Nothing is actually new. It turns out Jim Griffin reported this now almost 30 years ago in 1982, or 40 years ago in 1982, when he said that, some of these chemotherapies can cause some differentiation of leukemia cells. This absolutely has to do with nucleotide levels, because we can completely rescue these effects just by giving back the nucleotide that's not already there. So if we inhibit pyrimidine synthesis, we can rescue with uridine. We inhibit purine synthesis, we can rescue with hypoxanthine. So this is purely about nucleotide levels. You perturb nucleotide levels in the right way, cells will turn on these cell state changes. Well, a clue to what might be going on actually came from a former graduate student, Franny Deal, who recently graduated. She's now doing her postdoc with Rachel Green. But basically, Franny was studying some of these other things I was talking about earlier and rescuing cells with lots of nucleotides. And she made what we couldn't decide at the time was an interesting observation or not. And basically what she found is that if she rescued with too high a dose of specific nucleotides, it suppressed the ability of cells to grow. Now, this is probably how double thymidine block works, but she found in the model she was looking at that this occurred with very low levels of guanine, but it turns out different cells are differentially sensitive to the nucleotides they give them to suppress cell growth. And why this actually works is as follows, and that is that cells will always prefer to salvage what you give them. And so we can do fancy tracing experiments, and I don't have time to go into the details here, although I'm happy to talk about it, is if you give cells a lot of a single nucleotide, they will salvage it, and the feedbacks are such that you will basically inhibit all the other stuff that are made. And so effectively what this is it's a model of causing nucleotide imbalance, and how double thymidine block probably works is you cause an imbalance of thymidine to other nucleotides, you stall an early S phase, because it causes replication stress, and that replication stress is what synchronizes your cell cycle. But it turns out you can do this with any nucleotide, and imbalances can cause this in lots of different ways. And we think this is actually what is causing this differentiation response. That is, if we mess with nucleotide metabolism in any way, either artificially or with drugs, and it turns out maybe this even happens sometimes in normal S phase in the right environments, you basically have an imbalance in the amount of nucleotides that are required to synthesize your DNA. There's a known response to this, which is most people think of as the S phase DNA damage response. It's really the replication stress response. You turn on ATR signaling in check 1, and if this progresses, you can then get double-strand breaks with ATM in check 2. But effectively, what we see in all kinds of ways we test this, so for example, give a lot of guanine, give a lot of nucleotide synthesis inhibitor, we basically get check 1 turned on, and if this persists for too long, ultimately you turn on check 2. So this is basically causing replication stress, which led to the obvious hypothesis, turning on the replication stress response is somehow speaking to differentiation, probably through that signaling. Well, we don't think it has anything to do with the signaling. Well, the signaling we think is important to keep cells alive, okay? And I just want to show that this is really replication stress doing this. We can give afitacolin, which is a DNA synthesis inhibitor, and it will trigger differentiation in all kinds of leukemia cells and other cells at doses before it will cause cell death. And this is basically turns on the exact same replication stress response I just showed you. Well, how does this actually work? Well, I don't think the replication stress response allows cells to actually finish at some point replicating their DNA. It basically keeps cells alive and ultimately fixes the imbalance, but it is not what's causing the differentiation. Here's an experiment Franny did where she arrests cells with guanine. You get some DNA damage, but not all that much. She can inhibit ATR. The cells, are still growing, but if she inhibits ATR and then relieves their replication stress, they can no longer grow. You will basically recover if you don't have ATR inhibited, but if you do have ATR inhibited, you will no longer be able to grow. And the same thing in our AML differentiation. If we inhibit check 1, we will basically, this is a complicated graph here for the sake of time. I won't walk you through every single thing, but basically what this experiment is telling us is that check 1 inhibition does not prevent differentiation. It is just necessary to keep cells alive in order to differentiate. Well, this is actually quite exciting because it says somehow inducing replication stress is changing cell state. And so we can compare this in a system looking at Brequinar, so replication stress induction, with removing HOXA9 in that original expression. And it turns out these don't exactly get you to the same place in gene expression state. But in some ways, that makes sense. It turns out you can partially now begin to change your gene expression and your chromatin state along a pathway that moves you towards differentiation, but you can't go all the way because you cannot turn off the HOXA9 that is actually keeping these cells immortalized. And so what we think replication stress is ultimately doing is actually changing somehow the state of your gene expression that now allows you to do something different. This is completely dependent on known things we know about. So PU1 expression is necessary for myeloid differentiation. You get rid of this, you can't turn on lysozyme GFP and these other things, but something about replication stress is allowing you to turn on PU.1 expression that even allows you to do this thing. Now, I'll just tell you that this kind of challenges, at least the way I think about cell state change or differentiation at a very high level, and that is somehow cells make a decision in G1 to now enter this G0 differentiated state. But what this is telling us is that perhaps it's replication stress by moving through S phase that now gives you this a chance to change what genes you're actually expressing. And so is there any evidence that's here for this? And so we did a number of experiments to look at that. One of this is we can begin to look at, well, does this actually matter in S phase? And so we can use the so-called Fuji system, which marks cells in S phase by basically accumulation of this fluorescent M-cherry geminine construct. And so we can go back to the system, induce replication stress, and basically what we find is that cells will enter S phase, and while they're in S phase, they'll start to turn on lysozyme GFP, and then they'll progress through the cell cycle, really arguing that they're making this cell state change as they progress through S phase. We can do this with fancy single-cell seq experiments where we actually go through and actually can do single-cell seq at time points after Brequinar treatment. And what we find is where cells begin to turn on, say, PU.1 expression is right here at this 12 to 24-hour time point. And if we begin to map this on how this actually looks at the cell cycle, and in fact, we can actually do things like what Nicholas Raesky has done, where we can actually look at, is there some structure in the single-celled seq data that relates to something like cell cycle? And basically, the differentiation expression program tracks with the cell cycle, actually naturally lays out with tracking with the cell cycle, such that you begin to make all these changes with replication stress induction as you're going through S phase. This is not what you see if you turn off HOXA9, which we can do in this artificial system. That looks nothing like that. That just changes gene expression because you turned off this lineage-specific transcription factor. And so we really do not know how to understand this. I think what it is, telling us is that perhaps one way metabolism talks to cell state is that it messes with nucleotide levels. And if you mess with nucleotide levels such that you enter S phase and begin to have replication stress, you now change the ability to express chromatin differently at the loci where you're experiencing the replication stress. Lots of details to work out how this works, but why I find this model so attractive is that I like to remind people that if you ask what the origin of replication is, that is, where do you replicate DNA in mammalian cells, it's different for all cell types. You begin to replicate DNA at the open chromatin first. And Franny did some experiments to say that you get replication stress early in S phase because you enter S phase and then use a replication stress response to try to figure out and rebalance your chromatin. But perhaps this now creates a model where cells actually start at their open chromatin. And if they have the right metabolic changes that cause replication stress, it now allows you to work with the chromatin that's already open to begin to change what is the gene expression state. And that's going to be different based on your cell type, and perhaps as the beginnings of a way that one might begin to think about a new way that metabolism might be affecting cell state. Again, this is very early on. We haven't proven any of these things, but I find these ideas very attractive. They're things we're starting to think about. And I would love feedback from people who think about this in a very different way, because maybe we're just all crazy, but these are some of the results that we've seen. And so with that, I'm at the top of the hour. I'm happy to stay, like I said, as long as you want for questions. I tried to thank people along the way who did the work and our collaborators. Also want to thank our funding sources, and thank you guys again for your attention.
Thank you, Matt. Fantastic stuff. It is the top of the hour. We're formally complete. We understand that there are people who have appointments and need to go, but stick around for the questions and discussion. We have some great questions coming up. Matt, Ling Tao asked a question about Sharanya's experiments. He or she asked, how was the nutrient availability measured? Is it bulk tumor with tumor cells and other types of cells? And does it include extracellular nutrients?
Yeah, so the data I showed was just bulk tumors. It's just basically rip the tumor out of the mouse and do metabolomics on everything. So intracellular, extracellular, all the different cell types. And so look, I realize that's a very crude experiment, but it's still clustered in a way that was really interesting. I think if you want to get into finer resolution of metabolites, now you get into what I think is one of the biggest technical challenge that all of us who think about metabolism face. And that is, there's no good way to do single cell metabolomics. And that's simply because I can't isolate cells fast enough to actually say for certain I didn't change metabolism. Now, there's people who do this, and you certainly can learn from those experiments. And other parts of my lab has tried to come up with other tricks and ways that we can infer metabolism. But that is just bulk tumor metabolite levels. But I want to say we've done other things. We've done nutrient tracing. That looks, it's reading. It's a different readout. We've done different orthogonal readouts to look at metabolism. We've done gene expression and asked what the metabolic gene expression is. That was done a long time ago. We need to expand that to metastasis. So we're doing that now because there's clear predictions that would be made. But it is a shortcoming of our experiments, but unfortunately, I think the experiment you're alluding to that we should do, we're well aware of what the shortcoming is. I feel very strongly always to admit what we do and do not know, or what our shortcomings are. But I'm not sure it's possible to do the perfect experiment, at least with our current technological capabilities.
I had a quick question, Matt. When you talked about your really interesting experiments on caloric restriction and ketogenic diets with palm oil or with soybean oil, these experiments, I assume, were done in intact animals. Do you know, or do you have any idea whether, to what extent the immune response might actually be contributing to some of these effects?
Yeah, I was going to guess someone would ask that question because it comes up all the time. So
Of
So,
course.
so what I can tell you is that all those experiments were done basically with transplant models. We did them with transplant models of lung and pancreas cancer, all purebred, pure C57 black 6. So fully immune system intact mice. Okay. So without question, there clearly is something going on with some immune things, and immune things could very well be working there. We have not asked those questions. We need to ask those questions because it comes up all the time. Although there is something a bit paradoxical about this, because I'm sure many people have noticed, and that is I always find that the tumor take rate is better if I do a syngeneic transplant than if I do even into a nude mouse transplant, which is actually the opposite. Most people usually look for, you go into an immunocompromised mouse and look at things, but we typically find the take rate is better. So while there might be some immune interactions going on, I think there's also something else about maybe immune positively helping or not helping as well. Like it's not just tumor control, but I think there might be some other, must be adaptive immunity, because that's what is different between these, at least most of the models that people use for the immunocompromised thing for tumor take rate. But I think it's a really interesting question and something we need to think about more. And if other people are thinking about it and want to talk to us about it, we certainly are very interested in it, because even though I'm an immunologist by training, I don't like to even admit that very often, because I was trained in immunology before like even suppressor T cells were, they argued to exist, and tumor immunology was like, they laughed at you if you thought about tumor immunology. So a very different era.
Absolutely. Charlie Roberts has an interesting question. He says, you beautifully demonstrated that caloric restriction can have negative impacts upon cancer's growth in mice. Cancer-associated cachexia in people is often characterized as a toxic side effect of cancer. Rather than being maladaptive, is it possible that cachexia is an evolutionary selected host response designed to deprive cancers of nutrients in order to slow or block growth? With this in mind, have you evaluated the extent of caloric restriction upon tumor growth to determine whether there is a caloric level at which mice survive but the cancer stops growing or dies?
Yeah, so Charlie, that is a fantastic question. And in fact, I want to allude to some work that's a different talk, but it's work we published a while ago, actually looking at tissue wasting in pancreas cancer. And basically, we found that in pancreas cancer, in the pancreas, one of the causes of tissue wasting is actually your inability to digest food. Remember, pancreatic enzymes allow you to digest food. And so therefore, we could reverse that. And when we reverse that, the mice died faster, which goes along with exactly what you're saying, that perhaps sometimes, maybe starvation or cachexia or some kind of tissue wasting could actually be an adaptive response, meaning good for the host because it allows the mouse to live longer. We worry about that a lot, but I want to come back to, and I think it's going to depend, and we've actually continued to work in that area. And I think without getting into the details, because this is, it's still work in progress, and I'm not 100% confident in all the stuff that we've found so far. But what might be coming together is it actually probably depends on the specific nutrients. So getting back to what I said before, I don't think it's so much as, being cachectic is necessarily good or bad, but it might be what's actually driving you to that state, about whether or not that's bad for the tumor or not bad for the tumor, right? How that actually interacts with the tumor. So I think there might be situations where you actually might live longer. And in fact, patients are very bothered by this. And it's a whole different thing when talking about patients. And I never give advice based on animal studies to patients when it comes to diet. I usually just say we don't know that. But it does at least raise an interesting possibility, because patients are often very worried about being, their family members not eating. And of course, anyone who's taking care of patients knows that there's nothing you can really do about that. But it is maybe at least comforting to know that maybe that is at times could actually be an adaptive response, because there is evidence, at least in animal models, that maybe that might actually help you hang on a little bit longer. Now, the flip side of that is, though, one has to also be very careful interpreting these studies, because, there's sort of two issues with tissue wasting or cachexia in humans. One is, in an untreated person, how does that actually affect their progression of the tumor or their overall survival? But another one is, how does that affect your ability to actually give drugs? And as an oncologist, if people are in too bad a shape, you actually can't give them the therapy they need. And so if you believe our therapies are effective, it actually now gets more complicated in other ways. So I think these are fascinating and important questions to think about. And definitely we are thinking about them. And I guess I would encourage more people to think about these things, because there is sort of this thing that at least I learned in clinical training, cachexia bad, you always want to reverse it. And you can't always do it. And I think there might be a more nuanced answer to that, which
Yes.
is inherent in your question, Charlie, and something we're really interested in unpacking further exactly along the lines of what you're asking.
And this is definitely an area that overlaps with infectious disease and bacterial infection, certainly. Ronald Falgreeder, apologies if I've mispronounced your name. You mentioned that glucose uptake in cancer is saturated by approximately 1 millimolar glucose, and that serum glucose levels are much higher than 1 millimolar. However, isn't the tumor experiencing much less, even micromolar levels, glucose availability in the hypoxic regions of tumors than in the serum? Therefore, tumors or certain hypoxic regions of tumors may be experiencing glucose levels that are below the uptake saturation of 1 millimolar. Yep. Curious if you have any data on this.
Yeah. So it's a great question. I'm really glad you asked it, because it gives me a chance to talk about what people say and what there is actually data for. Okay. So I agree with you. It's an attractive idea that particularly hypoxic regions of tumors, they're not going to have blood, they're not going to have other nutrients, glucose could be depleted. And I don't know anyone who can actually measure in a regional way what local glucose depletion are. So, but it is likely that you do get certain regions of tumors where glucose uptake might actually fall below what the KM for uptake is, which is actually high hundreds of micromolar. We've done this. So we've measured glucose levels in interstitial fluid. We've isolated from mice. And this has the caveat of it's bulk tumor measurements. We can't isolate interstitial fluid from too small an area of a tumor, right? We need enough material to actually isolate interstitial fluid. But where we've measured it, even in pancreas cancer, in the pancreas, which is usually argued to be a particularly nutrient-deprived tumors, one of the big surprises was we were still measuring at least an average tumor interstitial fluid glucose concentration that was around 1 millimolar. And remember, if the KM for most of the glucose transporters, which are all passive, so how much glucose uptake you have really depends on what's outside the cell, is actually 500, 600, 700 micromolar. And so you're still above that, and you have to get quite a bit lower than that. So I don't actually know of any evidence that says you really get down into the 100 micromolar or less, even though there's a lot of people who make that assumption. Now, I'll just come back to, I suspect there are cells that are truly glucose starved. But again, I'll also come back to FDG PET scan works, and FDG PET scan ain't working unless there's actually glucose there to be taken up and light up a tumor. So regionally, it may be the case, but at a bulk level, that doesn't mean glucose is actually depleted. And so I think it depends on the question you're asking. If you're saying the response has to entail a whole tumor, I don't know of any evidence to say at a whole tumor level that you're really super depleted. But locally, you very well might be, and this might matter for some of, say, the immune findings where glucose, beautiful work from Sue Keck and others that really argue competition for glucose locally might affect some adaptive T cell responses, for example. Like, I don't think any data that we have speaks against that, but I do think one has to be careful with just the assumption that cells are hypoxic and therefore they have nothing.
These questions you answered. I'm going to skip down, and apologies to people if I've skipped your question and you feel it hasn't been answered. Chuck Shear has said, senescent cells which have durably withdrawn from the cell cycle into a G1-like state are abnormally large, and it has been reported that rapamycin can inhibit senescence induction. That's the inhibition of the TORC1 complex. Have you any idea as to how metabolism, protein synthesis, might determine the senescent phase?
Yeah, that's a great question, Chuck. And I wish you were a reviewer for Franny's paper where we're trying to report some of her findings, because basically what we find is that causing these things that cause nucleoside imbalances basically allow that those are sensed because you enter S phase, you get replication stress and you end up arresting, but the cells actually keep growing really, really large. And in fact, is a literature out there that an inducer of cell senescence is actually things that induce replication stress. Senescence is, many things can induce senescence, but there is a literature there around that. And we see exactly the same thing. mTOR inhibitors are upstream of all that because mTOR inhibitors clearly cause a G1 arrest, as most metabolic perturbations do. So most things that are sensed about metabolism, mTOR being the classic example, arrest you in G1, so you never even get to try to replicate, so you can never really get big. And so Franny did some interesting experiments where you actually can't give once you arrest in because of replication stress in S phase and keep growing really big, you can give mTOR inhibitors and that may stop you from growing further, but it doesn't necessarily shrink you. And so maybe that begins to speak with this. And it at least led us to begin to think about maybe some of the same perturbations that are causing the cell state changes we're looking at might also be part of at least some aspect of at least some of these senescent programs people are looking at. But clearly what Franny found is that replication stress inhibition completely decouples, or the nucleotide imbalance causing replication stress completely decouples cell growth from proliferation. And again, that has also been observed before. If you go back to the original cell cycle studies in yeast, there's a lot of yeast cell cycle mutants that basically allow you to get really big cells, but they arrest, and they almost all arrest in S phase, and they almost all look very much like this. So I suspect maybe there is something deeper going on here, but one has to now really speculate a lot. But if shifts in nucleotide levels causing this imbalance is somehow affecting these downstream things, maybe that is a tie to that observation about senescence. And I absolutely love that idea. And you could have come to our lab meeting 3 hours ago and had this exact same discussion. So.
Chuck always has really insightful comments. I'm going to combine several questions. And again, apologies if I'm misstating your question. There's a number of questions around hypoxia and caloric restriction, fatty acid synthesis. We know from work from Ralph de Bardinis and others that under hypoxia, you can still engage fatty acid oxidation by reverse carboxylation. Do you have any insights into these therapies under conditions of hypoxia?
Yeah, so there's a number of ways I can answer, or that the directions that question could speak to. I'll speak to a couple. So because you brought up Ralph de Bardinis's work, basically what they showed is that under hypoxia or electron transport inhibition, you can now switch to reductive carboxylation, and we did some work along those lines as well. But I think what has always been a mystery there is that even when this happens, if you look at the absolute rate of fatty acid synthesis, you're actually much lower, even though you're now doing it by this reductive pathway. And in fact, there's been a longstanding debate about whether or not that is even real. I think work that Christian Metallo did actually showed quite well that there is actually true net fatty acid synthesis via reductive carboxylation, but it's nowhere near what you see with under normoxia and glucose metabolism. And it's also been known forever that hypoxia will limit fatty acid synthesis in lots of different ways. And people often have assumed that this is an ATP problem or an oxygen limitation for fatty acid desaturation. What we think might be going on is we think that, all of this might be manifestations, though, of going back to the thing I talked about in the middle about if cells are electron acceptor inhibited, that is, they don't have enough oxygen, they can't run their electron transport chain, whatever, they are now in a more reduced state. And we actually think it's the more reduced state that ultimately is driving the reductive carboxylation. But because that is still gated by some oxidation reactions, it puts a cap on how much you can actually do. And I like this because it actually describes why you would have lower rates of synthesis, even though this might actually be better for you. The other piece of evidence that suggests that is true is that it's also been shown that acetate uptake can help you do fatty acid synthesis and hypoxia. And acetate uptake is downstream of all the redox steps. And that also is upstream of the desaturation step. So I think the fact that acetate can largely rescue the effects of hypoxia on lipid synthesis fits also with some of these models. And that's a very long-winded answer. And if you're an expert in the field, hopefully you followed it. If you're not an expert in the field, we do have a bioRxiv preprint on this. It's actually gotten much better that we're working on, but you can look that up. The first author is Lee et al., L-I et al. It's on bioRxiv if you want to read more about some of our data around this.
Thanks, Matt. I'll with a number of people asked, have you done some restriction or fasting versus periodic chemotherapy? Can you toggle these?
Yeah. So I'm sure where these questions are coming from, just to make sure everyone's on the same page, is, Walter Longo has done some very nice work, really arguing that fasting around the time of chemotherapy can actually be protective, more as a protective measure for host tissues than really enhancing the effect of the chemotherapy itself. But it also goes back to, if our therapies are working and you can't give the therapy, well, then you're not going to get the benefit, right? So if you can protect the host tissues, you're better able to give the therapy. I should say that the human clinical trials of this have been equivocal at best. There was actually the larger trial in breast cancer that was negative, that was actually quite recent. But nonetheless, this is a really interesting idea. It's not one that we have looked at specifically, but I guess I'll just speak to how we're thinking about the question. And I think one has to really separate what is, because I think this is the problem with diet and cancer, is people often conflate all kinds of different things into the same phenomena. And I think there's the question we've really been focused on is, how does the diet actually affect the nutrient availability and the growth of the tumor? And that is one axis. But then there's a separate axis, is how does the diet actually affect nutrient availability and how that might change response to therapy? And then there's even a third axis, which is how might that actually affect the host tissue response to therapy? And I think the hypothesis that is being asked in the question really has to do with that third one, and the questions we've been asking is the first one. But they're all good questions, and they're ones we're starting to think about as well, at least along the middle piece there, which is about how does diet maybe change your response to therapy. But I guess I'll just answer that question, as I can't answer it because we don't have data to speak about it ourselves. So the only data I know is what I referred to. But it does say that I think it is super important that you need to actually think about these things in terms of what question are you asking. And a specific diet could have different effects, and maybe it works great in one thing versus another, which actually goes back to Charlie Roberts' question. Is cachexia good or bad for you? And it, maybe it's good for you in terms of it makes your tumor go slower, but now I can't give you therapy, so it's bad for you. And then which of those is the winner is a very different thing.
One quick follow-up from Chuck Shear. Cells somehow know how large they should be even during proliferative cycles. Does replication stress uncouple cell growth mass from proliferation? Great question, Chuck.
Yeah, it is a great question. And Franny looked at exactly this question, and it absolutely does. So cells will get bigger and bigger and bigger. They actually do so in a balanced way, meaning their density doesn't change, their rate of protein synthesis doesn't change, their relative protein synthesis to other things doesn't change, all those things. I think you can get to be a large enough size. My colleague Angelica Amon, who passed away years ago, said if you get to a certain big size, you actually do get some cytosolic uncoupling of ratios of things being off. But we don't see that actually, at least to very large sizes. And Franny did an interesting experiment where she actually made cells very big by basically causing replication stress with nucleoside addition, and then could reverse it and looked at how they recover. And it turns out it's actually the biggest cells actually never recover. It's actually only the smaller cells can do this, but you actually probably now divide back to the same size you want to be and re-enter the cell cycle. So the mean cell population will remain high for a while, but that's only because I think once you get so big, you just never recover. But so the short answer is yes, it does uncouple this whole how big cells should know to be. But if they can actually recover from this problem, then they go back to knowing how big they should be, which is really interesting. And how cells know how big they should be I don't think is really a question that our data speaks to, other than more evidence that they seem to know and care what size they should be.
There's a great opportunity. You're fortunate at MIT to have Scott Manolis there.
Yeah,
and he's done incredible work on single cell measurements of cell mass and proliferation.
Yeah, in fact, it was Scott that allows us working with this guy, Teemu Miettinen, in Scott's lab, who's like a cell cycle guy who's looked at this, and really the experiments I just described from Franny all were in collaboration with Scott and Teemu. We wouldn't have been able to do them without them.
Wonderful. I am going to applaud for all of us to thank Matt for this amazing talk. We still have quite a large number of people on the call. Please join us next week for the inaugural Rising Stars of Cancer Research lectures in the Science of Childhood Cancer series. The event will feature thematically related talks on cancer immunotherapy by Yvonne Chen of UCLA and Sar Gill of the University of Pennsylvania. Join us then. And Matt, thanks again for this fabulous talk. I know there's a lot more we can be talking about. Thanks everybody for participating, and we'll see you all next week.
Thank you so much, Doug.